Hi students, welcome to SPSVERSITY. Today we are going to discuss about excretory products and their elimination. What are these excretory products? Due to different metabolic activities, so in our body, there are different metabolic activities are going on. And due to these different metabolic activities, so waste is produced. And this waste should be sent out frequently so that our body remains healthy. Otherwise, this waste may become toxic. So depending upon the habitat of the organism, there are different types of excretory products. Like some organisms will excrete ammonia as an excretory product. So that type of organisms are called aminotelic organisms. Like aquatic organisms like fishes, this one. Ureotelic, which will excrete urea as an excretory product, like terrestrial organisms. Uricotelic, which excrete uric acid as an excretory product, like birds, reptiles, snails, etc. So these are a few types of common excretory products seen in living organisms, like ammonia, urea, uric acid. And depending upon their habitat and their complexity of body organization, so the different types of excretory organs are there. So all living organisms doesn't have the same type of excretory organ. Like if you take invertebrates, they have tubular structures. Prawns, they have antennal glands as their excretory organs. Liver fluke and flatworms, they have flame cells as their excretory organs. Insects like cockroaches, they have Malpighian tubules as their excretory organs. So like this, the cockroaches have Malpighian tubules. Amphioxus has protonephridia as excretory organ. Earthworm, nephridium. Vertebrates, kidney. Like humans, you have kidneys as an excretory organ. So depending upon the habitat, the excretory product will be differing. So ammonia requires more amount of water. Urea requires still less amount of water. Uric acid requires still less amount of water. So depending upon their habitat and the availability of water, the excretory products will differ and the excretory organs will differ. Now we will discuss about human excretory system. In this human excretory system, kidneys are the excretory organs which regulate the amount of water, salts and other substances in the blood, which regulate osmoregulation. So the kidneys are bean-shaped structures, 10 to 12 centimeters long, 5 to 7 centimeters in width and 2 to 3 centimeters in thickness. So these kidneys will remove nitrogenous waste, urine and excess of salts from the body. Okay. From kidneys, there are tube-like structures that from each kidney tube-like structure arises, that is ureters. So from pelvis of the kidneys, the ureters will arise and they will join with urinary bladder. Urinary bladder, it is temporarily which stores urine to its maximum capacity, and it is released from the body through urethra. So urethra is a tube that carries urine from urinary bladder to outside of the body. So the end of urethra is controlled by a circular muscle. So that circular muscle is called sphincter. So here you can see the diagram of human excretory system. There are a pair of kidneys. From each kidney there is a tube-like structure arising, that is ureter. Opens into a urinary bladder. And below this urinary bladder there is a tube-like structure through which urine is sent out. It is called urethra. So there are a pair of kidneys. From each kidney one ureter, that is a pair of ureter, one urinary bladder and one urethra. And the blood supply, that is the renal vein and renal artery to kidneys, that is the right kidney and the left kidney. So there is a blood supply through renal artery, and blood is taken away from the kidneys through renal veins. Okay. So this is posterior vena cava, which will collect blood from the kidneys. And this is aorta, which will supply blood to the kidneys. So posterior vena cava will take out blood and aorta will send blood to the kidneys. Okay. So here kidneys play an important role in the excretion of human beings. So each kidney is composed of 3 parts. So when you go into deep into the parts of kidneys, so the outer region is called cortex, and the middle part is called medulla, and the hollow inner part is called pelvis. Okay. So the cortex is where the blood is filtered. The medulla contains collecting ducts which carry filtrate to pelvis. So pelvis is an hollow cavity where urine accumulates and it is sent into ureters. Okay. So the kidney can be divided into 3 parts: cortex, medulla, pelvis. Where cortex will filter the blood, medulla will carry out the filtrate to the pelvis. From pelvis the urine is sent out through ureter. So here the function of kidneys is to filter the blood to remove cellular waste products formed from the body. So at any given time, if you take 20% of blood is in kidneys. So humans can function with 1 kidney. If one ceases to work, the other increases his size to handle the workload. So humans can have a normal functioning of excretion with 1 kidney. Okay. The kidney has other functions also. Okay. But it is usually associated with excretion of cellular waste, like urea, which is a nitrogenous waste produced from liver from the breakdown of protein. It is the main component of urine. Uric acid, which is usually produced by breakdown of DNA or RNA. Creatinine, which is produced as a waste product of muscle action. So the urea comes from breakdown of protein, uric acid from DNA or RNA, creatine from waste product of muscle action. So all these compounds have nitrogen as a major component. Whether urine, urea, uric acid, creatine has a nitrogen. Okay. So they are excretory organs as well as they are one major homeostatic organs which maintain internal conditions. Homeostatic means which maintain internal conditions as one of them. So they control water pH. Not only excretion, they control the pH. Secrete an hormone that is erythropoietin, which stimulates red blood cell production. And activate vitamin D production in skin. That's the reason why doctors give so much of importance to urine sample. That is, whenever it is diagnosed by disease, that is urine sample. So here you can see the parts of a kidney. This inside kidney, if you see the parts, the outer region is called cortex. The middle one is medulla. And there is a pelvis. This is pelvis, into which the urine is collected and drained out from pelvis to ureter. And through this there is a renal artery and renal vein, where renal artery will send blood from renal vein, the blood is collected from kidneys. And the kidney is well protected with layers. The kidney is well protected with layers. Okay. So in this, if you see carefully in these parts, along with the cortex, in the cortex and medulla, there are nephrons. What is this nephrons? So these nephrons play an important role in functioning of kidneys. It is the filtering units of kidneys are called nephrons, and there are approximately 1 million nephrons in each kidney. There are approximately 1 million nephrons in each kidney. And these nephrons are located in cortex and middle of each kidney. So each kidney has 1 million nephrons. Okay. And these tubes of nephrons are collected by surrounded by cells and network of blood vessels. Therefore, whatever the material leaves the nephron, it will enter into the surrounding cells, into the bloodstream network of vessels. Okay. And these nephrons are structural and functional units of kidneys. So the function of kidneys is actually performed by nephrons, where they will do the filtering of the blood. Now we will discuss about the parts of nephron. What are the different parts of the nephron? How they will perform the function of filtering. So each nephron consists of following parts. That is, glomerulus, which is a network of blood vessels. Bowman's capsule. Proximal convoluted tubule, which is called in short form PCT. Loop of Henle. Distal convoluted tubule, which in short form it is called DCT. and its last one is collecting duct. So nephron has glomerulus, Bowman's capsule, proximal connected tubule, loop of Henle, distal connected tubule, collecting duct. So now we will come into the anatomical unit, that is uriniferous tubule, which is nothing but nephron. Okay. So in nephron there are different parts, like that is the renal corpus, that is the glomerulus, the Bowman's capsule, PCT, loop of Henle, distal connected tubule. So this distal connected tubule, this is when you come to renal corpuscle, which is called combinedly the Bowman's capsule and the glomerulus is combinedly called renal corpuscle, which is present in cortex. Okay. So this glomerulus is a tuft of blood vessels, and Bowman's capsule is a cup-like structure which will hold these tuft of capillaries. They are present in cortex. Next, tubular section, that is proximal connected tubule, loop of Henle, distal connected tubule, and collecting duct. So like this we can differentiate the parts of a nephron, that is renal corpuscle, tubular section, and collecting duct. So renal corpuscle, which consists of, that is glomerulus and Bowman's capsule. Tubular section, which consists of, that is proximal connected tubule, loop of Henle, distal connected tubule. And next one is the collecting duct. Okay. So whatever the materials which are collected, that is filtered by blood through these nephrons. Okay. So whatever the blood stream, the water, glucose, amino acids, and many salts are reabsorbed. That is, during the process of filtration of blood, this water, glucose, amino acids, and salts are reabsorbed by different process like osmosis, diffusion, and active transport. Okay. Like water by osmosis, diffusion by glucose, active transport of amino acids, where ions are from filtrate to surrounding cells. Okay. So small villi-like projections will help in active transport of glucose out of filtrate and speed up of reabsorption. So from these again the components will return to bloodstream. That means whatever the materials are needed for, whatever the materials needed for our body, again they are reabsorbed from the body, from the kidney, that is the nephrons, by various processes like osmosis, diffusion, active transport, like this. Okay. So whatever it is, again they return to bloodstream. So the filtrate reaches to the endoproximal tube. So whatever it is filtered, that is which we call urine, which will reach to proximal tube, and the fluid is isotonic with the surrounding cells. So glucose and amino acids are removed from the filtrate. Then the filtrate moves into loop of Henle, where water is removed from the filtrate by osmosis. Even not only glucose, amino acids, even water is removed from the filtrate by osmosis. Okay. And along with this, the glucose, the what we call the amino acids, water, along with this, even minerals are also reabsorbed. Like When the filtrate is passing through loop of Henle, there is an increase in sodium concentration in the cells. That is, as you move from the area of the cortex to inner pelvis of the tubule. So this causes water to be drawn from the filtrate. Okay. So these high levels of sodium are a result of active transport and results in increased concentration of filtrate due to water loss. Due to water loss, what happens? The filtrate concentration will increase. The filtrate will become highly concentrated. And the surrounding cells, as they take water, they become diluted, that is, they less concentrated. Okay. So here in this picture, you can see the different parts of the nephron. Like you can see this is glomerulus, the tuft of capillaries. This is Bowman's capsule. Okay. Next, the renal tubule, the proximal connected tubule. The next one is the loop of Henle, the distal connected tubule, and collecting that. Okay. So here, the blood vessels which carry blood to the nephrons, that is, they are called efferent arterioles, which take blood towards these nephrons. And after filtration, whatever the blood is sent out, that is efferent arterioles. That is efferent arterioles. And this process which happens, that is, whatever the blood is filtered from glomerulus and it reaches to the what we call tubular renal tubule. So this process is called glomerular filtration, which happens in glomerulus and Bowman's capsule. And whatever the filtrate passes through this renal tubule, so from this filtrate, the nutrients, water and essential ions are again reabsorbed. That is called reabsorption. The first step is filtration. The second step is reabsorption, which takes place in tubular region. Next is secretion, where when the filtrate is moving through this tubular part, undesirable molecules are sent into this from the blood, like urea. These all are sent into this. Secretion takes place from peritubular capillaries. So in nephron, filtration, reabsorption and secretion takes place in a sequential manner. That is, glomerular filtration, tubular reabsorption and tubular secretion. So these are three key functions taking place in the what we call in nephron: glomerular filtration, tubular reabsorption and tubular secretion. So these are the different parts which are present here. You can see in cortex, that is renal corpuscle. This entire part comes under renal corpuscle. So where it consists of glomerulus, the network of blood vessels, it is called glomerulus, which holds the tuft of capillaries, the Bowman's capsule. And the remaining part comes under, that is renal tubule. Here it is proximal convoluted tubule. So proximal convoluted tubule, which is in short form, it is called PCT, which are present in cortex. So in the medulla there is loop of Henle, which is present in medulla. Next, that is the collecting, that is the distal convoluted tubule, which is called DCT, which opens into the collecting duct. And all the parts of this nephron are connected by, that is, the peritubular capillaries. That is, each and every part of this is surrounded by, that is, peritubular capillaries. The blood vessels which surround the parts of nephron, they are called peritubular capillaries. So here you can see the different parts, like the branch of renal artery which supplies blood. This is efferent arteriole which supplies blood to where blood enters. This is efferent arteriole through which blood is sent out, filtered blood. Glomerulus, Bowman's capsule, proximal convoluted tubule. These are blood capillaries which are surrounding. This U-shaped part is called loop of Henle, and this is distal convoluted tubule, and this is the collecting duct where the distal convoluted tubule will open. So these are different parts of the nephron where we can see, that is, the filtration in renal capsule here. The reabsorption and secretion which takes place, that is tubular reabsorption, tubular secretion takes place in the tubular region of the nephron. So we'll see how the filtration of blood takes place, what actually takes place in nephron. So whatever the blood enters the glomerulus, that is, the blood enters the glomerulus through renal artery. It enters the blood through renal artery. So whatever the blood enters, the blood is under high pressure. It enters with a high pressure. That is, through efferent arteriole, the blood enters through high pressure. As the blood enters with high pressure, the capillary walls are very thick. These capillary walls are 1 cell thick. Okay. So here you can see, that is, this is efferent arteriole. This is efferent arteriole through which blood leaves, through which blood enters. Between this tuft of capillaries, there are cells. These are called mesangial cells. And this tuft of capillaries, which is surrounded by Bowman's capsule. So between Bowman's capsule and the glomerulus, that is, there are cells, that is, the podocytes. Okay. So whatever the blood enters with high pressure, so this plasma filters the membrane under high pressure where proteins cannot pass through these fenestrations, that is, openings. So here in this diagram we can see clearly the layers of Bowman's capsule. So these are layers of Bowman's capsule. So which holds this tuft of capillaries. So this is glomerulus with a tuft of capillaries. In between glomerulus there are mesangial cells. Okay. So here whatever the capillaries, the glomerulus which is made up of tuft of capillaries, whatever the blood is filtered from the glomerulus. So the filtration membrane is held in place by specialized cells. They are called podocytes. So in the picture you can see there are specialized cells. So these are called podocytes. So these podocytes, through this podocyte, this blood is filtered. So glomerulus is a mass of thin walled of capillaries. And when you come to Bowman's capsule, it is a double walled cup shaped structure. From this Bowman's capsule, it leads to the tubular part, that is proximal tubule, a loop of Henle, and last, that is distal tubule, that is which extends into medulla. And distal tubule connects the loop of Henle to the collecting duct. Okay. So here, this all part, that is loop of Henle, is present in medulla along with peritubular capillaries. This peritubular capillaries which surround loop of Henle, it is called vasa recta. And the remaining parts, that is the glomerulus, the Bowman's capsule, the proximal connected tubule, distal connected tubule, these all are present in cortex, which is surrounded by the blood vessels. So there is a specific name, the blood vessel which surrounds the loop of Henle, that is vasa recta, which is nothing but the what we call the blood vessel which is surrounding the loop of Henle. Okay. So here you can see the cortical circulation. Okay. So this is glomerulus. Each one is one glomerulus. So efferent arteriole, efferent arteriole, and this is the cortex region. Okay. So the medulla, which are the nephrons which are present in cortex, they are called cortical nephrons, and which are present in medulla, they are called juxtamedullary nephrons. Okay. So here you can see, this is a renal pyramid, which is the internal part of a kidney, where it is surrounded by intralobular vein, intralobular artery, and below this there is a minor calyx, which opens into the big one, that is the calyx. Okay. So this is what is seen in cortical circulation. Okay. And this is the different parts of a nephron: glomerulus, the Bowman's capsule, the proximal connected tubule, loop of Henle, this is vasa recta, distal tubule, distal connected tubule, and collecting duct. Okay. So here you'll see each and every part function. So first we'll come to the function of the tubules. What actually happens in the tubule, that is proximal connected tubule and loop of Henle, distal connected tubule, what happens? Initially we'll start our discussion with proximal connected tubule. Follow us on social media, SPSVERSITY. So PCT is lined by simple cuboidal epithelium, that is, which is nothing but simple cuboidal brush bordered epithelium, which will increase the surface area of absorption, that is for reabsorption of minerals, the nutrients, the water and glucose, amino acids. So most of the essential nutrients, 70 to 80% of nutrients like electrolytes, water are reabsorbed in proximal connection, which will help in maintaining the pH and ionic balance of the body fluids. Not only reabsorption, even you can see secretion to balance the body fluid like hydrogen ions, ammonia and potassium ions, filtrate and by absorption they are secreted into it. That is, 70 to 80% of nutrients are absorbed in proximal connected tubule, along with there is secretion of this like hydrogen ions, ammonium, potassium ions into the filtrate. So these are secreted into and these are reabsorbed. There is glucose, water, electrolytes that are reabsorbed. So here in proximal connected tubule, you can see both the reabsorption and tubular reabsorption and tubular secretion takes place. So tubular reabsorption where 70 to maximum takes place in proximal connected tubule. You can see here the nephron. So this is proximal connected tubule, so which is freely permeable to water. The proximal connected tubule and the descending limb of the loop of Henle is freely permeable to water. That means water can be reabsorbed. So where water can be reabsorbed. Whereas when you come to ascending limb of loop of Henle, this ascending limb where it is impermeable to water, where water absorption is not seen. And here you can see this is when you come to distal collecting tubule, that is DCT and collecting duct. So in this DCT and collecting duct, there is a variability of permeability of water. That is depending upon the need and the hormones secreted, this distal connected tubule and collecting duct, the permeability of water will differ. So that means in proximal connected tubule and descending limb of loop of Henle, the water can be reabsorbed. Whereas in ascending limb of loop of Henle, the water is impermeable to water. Water cannot be reabsorbed. Distal connected tubule and collecting duct where the permeability of water will be differing depending upon the need of water and amount of water in the body. And hormones also play a major role in this. You can see the nephron osmoregulation, where in proximal connected tubule we can see the reabsorption of water by osmosis. Whereas in Bowman's capsule you can see ultrafiltration under pressure. The blood enters into with high pressure, which is called GFR, glomerular filtration rate, which will decide the filtration, the functioning of the nephrons. So here water is reabsorbed by passive osmosis. Passive osmosis means no energy is not required. So for absorption of water, energy is not required. That is, osmosis is an passive process where the water is reabsorbed. And when you come to the, that is here and here, there is PCT and descending limb of loop of Henle, the water reabsorption takes place. And here it is impermeable to water, there is no reabsorption of water. And here you can see variable permeability of water. Okay. So here the water is reabsorbed, and here you can see in the middle lobe region the water is reabsorbed, and sodium ions are secreted into this. Even in collecting duct also the water is reabsorbed. Okay. That means as you move from cortex to middle lobe, the salt concentration will increase. Okay. So this is collecting duct, this is DCT, this is ascending limb of loop of Henle, this is descending limb, and this is the PCT. So here you can see the more salt concentration here. So here also you can see the water reabsorption. Okay. So here loop of Henle, where in this the reabsorption is minimum. So this region plays a significant role in maintenance of high osmolarity and middle interstitial fluid. So here the descending limb of Henle is impermeable, that is, it is permeable to water, but almost impermeable to electrolytes. But quite opposite is ascending limb, where descending limb is impermeable to water but allows the transport of electrolytes actively or passively. Therefore, the concentrated filtrate pass upwards and gets diluted due to passage of electrolytes in the middle of the fluid. That means as it is moving through descending limb of Henle, as there is permeability of water, that is, there it becomes diluted, but here there is no permeability of water in ascending limb, that's why the concentration will increase. So here you can see at each and every part what are the nutrients are taken, what are the nutrients are secreted, that is where which are reabsorbed, which are secreted. In proximal connected tubule, it is bicarbonate ions HCO3, sodium chloride, water, nutrients like glucose, potassium are absorbed. That is reabsorption. Whereas the hydrogen ions that put on ammonia are secreted into this. Here you can see secretion. So this is loop of Henle, which is, this is called descending limb of Henle, where it is permeable to water. So there is water reabsorption takes place, but it is not permeable to electrolytes. So this is thick segment of a descending limb, this is thin segment of descending limb. So this is ascending limb, this entire part is ascending limb, where we can see the reabsorption of electrolytes like sodium chloride and urea is secreted into this. So the filtrate will pass in this direction. So it will pass in this direction. And then when it comes to distal convoluted tubule, where sodium chloride, water, bicarbonate ions are reabsorbed, and potassium and hydrogen ions are secreted into this. And even you can see the collecting duct where water is reabsorbed. So wherever the water is reabsorbed, the concentration of filtrate will increase. Okay. So wherever there is a secretion of this one, the concentration that is decrease. So in each and every part of this nephron, you can see the tubular secretion and you can see the tubular reabsorption. So in this part there is the glomerulus and Bowman's capsule. Only here you can see the ultrafiltration, where we can see the filtration of blood, the waste from this. Distal connected tubule, where you can see the reabsorption of sodium and water, as well as it is reabsorption of bicarbonate and selective secretion of hydrogen and potassium ions and ammonia into filtrate to maintain pH and sodium potassium balance. Here you can see there is an osmoregulation of, that is the regulation of water and the electrolytes. So like this, there is a regulation of the sodium and potassium balance, that is the osmolarity, which we call the osmotic concentration. Okay. Now we are going to discuss about an interesting one, that is counter current mechanism. So counter current means opposite. Here we are going to discuss about vasa recta and loop of Henle, which play an important role in concentrating the urine. That is, in loop of Henle there is a descending limb and ascending limb. So this counter current help in maintaining increasing osmolarity towards inner medulla. That is,